Welcome back, everybody, and welcome to the first full-length episode of Activated Sludge. We're a little foamier than normal in the aeration basin today, but it's okay. I'll explain what you're seeing and why there's a little bit of foam in there, and we'll go from there. But what you're looking at is the aeration basin. That's the mixed liquor suspended solids. But first, I kind of want to show you how my plant works from a bird's eye view. So we have our headworks over there. That tall old building, which is actually really cool. If you zoom in, you'll see the forms from like way back in the day when they formed it up. That's our primary clarifier. And then the water gravities over to this manifold over here, and we split flow between this EQ kind of is our biological process. EQ, the system kind of draws up and down on it throughout the day depending on flows. And so we'll either send to the trickling filter or to the membrane bioreactor depending on what processes we're running. We've got microscreens right here. So this is a form of preliminary treatment, but it's specific for this MBR because the MBR's pore sizes are so small that the tiniest stuff can blind them. So we go ahead and send it through some screening. From there, it drops into the anoxic zone, and I'll talk about that in a second. Aeration, membranes, and there's a RAS line. We're gonna go through all this here in a second. So let me get down on the catwalk a little lower and kind of show you an up-close view of that aeration basin and kind of talk about what's going on. Okay, so what you're seeing right over there is our anoxic zone. I'm not gonna get into that today because this is just an introduction to activated sludge. I really want to focus on aeration here. And what you're seeing with this foaming is actually, you shouldn't see this level of foaming in a conventional activated sludge plant where you're just doing BOD or TSS. What you're seeing here is the product of a low F to M ratio and a high MCRT, okay, and a really high MLSS. I run about an 8,000 MLSS. The thing about MBRs is that you have a lot of biomass in a small area, and you can treat a lot of water in a lot of wastewater in a very small area. Okay, but that comes with some of its, issues. And this level of foam is actually not concerning. If it was billowing up and pouring over, or if I had a clarifier and it was going under the baffles and over the weirs, that would be a definite problem. But I don't let perfect get in the way of good. I don't want that to be the enemy of good. And this is fine. It's more foam than I'd like, but I really want to get this video out to you guys, and I'm not going to wait for my aeration basin to look perfect. These are things you have to deal with. So this could be a few different things. It could be filaments, it could be surfactants, there could be a number of things this is. And when I get to the settleability test video, I'm going to show you how I determine what it is without having to take a foam sample and send it to the lab. So anyway, at the beginning, as the influent comes in, ignoring the anoxic zone, what's going to happen is we've got what these diffusers are supplying oxygen. We're going to go over this in the whiteboard a little later. They're supplying dissolved oxygen to the aeration basin, and these microorganisms are uptaking it. And remember, BOD is the biochemical oxygen demand that the wastewater would put on the environment. So what we've done is we've farmed, in essence, these microorganisms and they're going to consume that organic matter that has a BOD load to it. We're giving the dissolved oxygen to the microorganisms, and they exert the demand on this aeration basin, and they stabilize the wastewater, and they've eaten the food, okay? And so then they move over throughout. This is a plug flow, meaning it starts there and ends here, and it just kind of moves linearly. And then by the time we get here, and I'll talk about this on the whiteboard, the BOD is pretty much gone, and then it moves to the next process, which you're gonna hit in a second, and that's where we're gonna manage the TSS. So this is the mixed liquor suspended solids, which a high percentage of it is the volatile suspended solids, and that's kind of how an aeration basin works. If you have any questions, please comment. If you think I missed something on that one, of course, please comment down in the comment section. Now let's move on over to the membrane chamber. And so from aeration, what happens is we transfer over, and normally this be going over to a clarifier, but I've got membranes, so I'm gonna kind of explain what you're seeing here. This would act kind of like a stilling well would. You see a little bit of foam carryover. It's not a big deal. You can actually see it's not much foam given the level of the mixed liquor coming in. It's just kind of annoying. If it was to become a problem, I would have to deal with it, but it just hasn't. So these pumps in the bottom, they actually pump over to the membrane chambers. There's pipes on the outside of the structure, and they come up and they upflow, right? And so this is our return activated sludge right here. I'm going to go down below and show you those pipes in a second. So what's happening here with this blower that's constantly shaking up these membranes is we've got the spaghetti noodle style membranes, and so these are Suez, which I think actually Veolia just bought them out recently. But anyway, this air scour kind of knocks them around and makes sure that they don't blind due to the biology sticking to them, because there's a vacuum being pulled by the Permeate pumps to pull, this is the Permeate line right here, to pull that clean water out of there. And the flock in a normal clarifier would settle down, and we're going to talk about that on the whiteboard a little later. But in here I need good flock, because without good flock, my membranes would blind. And so what happens is we pull that really clean water off of the flock as it passes by, and the air scour keeps the flock moving along. And there's that beautiful RAS. Okay, so let's now go down below, and I'll show you my RAS line and my waste activated sludge pump. Okay, so now we're underneath the catwalk where I was just standing, and that little flow over, what I was calling the RAS, is the return activated sludge. It takes that 8-inch pipe and it heads back to the front of the plant. Okay, now we send it to an anoxic zone. You're going to see on the whiteboard in a minute. In a conventional plant, you would send it to the beginning of aeration. We are a different style. I will cover that in a different whiteboard segment at a later time. This is my WAS pump. It's a progressive cavity pump. I've talked about those. There's actually a video on progressive cavity pumps and how they work. I'll put it in the links below, so you can go check that out. But you see, we're pulling out of the same chamber, okay? And so this sends it to the digester for aerobic digestion. And the other thing that's interesting about this is I've got two separate lines. Sometimes you'll have, you use the same line and there'll be valving. So if the RAS line has a tap on it, you can just valve up those, the WAS line to just take a little bit off the RAS. And so the WAS is how we manage how much biomass we have in the plant. Okay, and over here we have our permeate pumps. They're pulling that water off. Again, this is a membrane bioreactor. It's a little different than conventional, but it's worth showing you. These are our effluent pumps that are pumping off. And then over here you'll see a couple chemical tanks. That's a sodium hypochlorite for our clean in place. And our citric is the blue for our clean in place as well. You typically have an acid and a base of some kind. Our base is sodium hypochlorite. We use it to burn off the biology off the filters. And then the citric is to descale. And soon I have to do a recovery clean. It's gonna be really great. I'll actually have one of the chambers empty at a time and doing an acid bath to it and a base bath. We're gonna fill up the chamber with a certain amount of chemical per the manufacturer's instructions, and I'll take you along with me for that. Something cool we're working on, like I said, is reclaimed water. And so over there, we actually pump right now to our contact chamber and mix effluents with the trickling filter, but soon a UV skid's going in over there, and we're going to be putting all that in. And you better believe I'm doing a video about that when we've got a UV skid and we can have two different kinds of disinfection here. So okay, well that pretty much sums up the membrane bioreactor, my little guy. It's tiny, but it's powerful. There are way more complicated versions than this, but it suits our need at our district, and it's a really, really great tool to use here. So let's head over to the whiteboard and we will talk about conventional activated sludge. Welcome to the whiteboard segment, everybody. I'm super happy to have you here. I've got a couple housekeeping items and then we're going to jump right into this lesson. First, if you're getting anything out of this video so far, please like the video, subscribe to the channel, and pass this stuff to your friends and colleagues, and let's help folks get certified. Second, I apologize for the crude nature of this image. I did try to edit it a little bit. ChatGPT made this for me. I want to avoid copyright issues with pulling stuff off the internet. You just never know who's out there looking to nail you for using their stuff off their website. So anyway, I asked ChatGPT to make it. It struggled immensely, and I made a little edit in Microsoft Paint to make it look make a little bit more sense, but it still looks, got these weird spots right here, and it's just, apologies for it. It will get better over time. I am no AV wizard, and I am no artist, or apparently struggle with drawing straight lines too. So anyway, the information here is solid, so let's just pay attention to the lesson. Please don't pay so much attention to my kindergarten art here. So, jumping into conventional activated sludge. We are, just for the people out there who are experts, we are talking about conventional activated sludge and plug flow, meaning the flow moves linear. We're going to get into the different styles of activated sludge, MLE, BART info, contact stabilization, all these other different ways that you can do activated sludge. And we're also going to get into the different styles of feeding it, step feed, complete mix, these types of things. But let's just keep it simple. I don't want to blow anybody's brains up as we're trying to just learn the conventional system. So you notice there's a clarifier here. This takes the place of my membrane chamber, okay? I have a membrane bioreactor. In my plant, we extract the water off the flock and as a really clear effluent. We call it permeate. And in a clarifier, there's liquid-solid separation, and the flock forms and drags everything down, and then you get this clear effluent on the top. We'll talk about that in a second. Okay, so this is also the secondary clarifier, and I'm going to make a point and say this just because I was told this when I was coming up, and it was incorrect information. This was like my third or fourth wastewater job, the chief plant operator told me, because we didn't have a primary clarifier, he said this was the primary clarifier because we didn't have one already. That's incorrect. This is the secondary clarifier, whether you have a primary clarifier or not. The secondary clarifier is referring to the process, not how many you have. You can have several secondaries, you can have several primaries. Primary treatment is different than secondary treatment. So I know I'm hitting it hard here, but the secondary clarifier is not referring to the sequence of clarifiers, it's referring to the process in which it's involved, okay? And by the way, when it comes to clarification, I will link to primary clarification part 2, where I actually tour my primary clarifier in the bottom of it, and the whole thing's drained and you can kind of see the anatomy of a clarifier. Secondary clarifiers can have different hardware. We're not going to get into that right now, but I'll also in the links below put a video where we worked on our secondary sedimentation basins, and I do a whiteboard segment where I kind of explain how those flights move under the water, because it's hard to see some of this stuff sometimes. So anyway, let's jump into actually the lesson. So this is a circular system, and it's hard to actually tell you where to start in a circle. So we're going to start at the beginning of the circle, which is the influent, or the primary clarifier. Effluent, whatever's coming into aeration, okay? This can be skip the primary, it can be screened and sent in. It will have a primary clarifier like I have, or I've seen it not screened and sent in, raw influent, just right into the aeration basin, and that's awful. The stuff, I'll keep it G-rated, but those of you who've worked at a headworks can imagine the things that would make it into the A-bays that would pass through a pump, and it was a lot of stuff floating around in there. So anyway, the influent is loaded with the food. This is the BOD, biochemical oxygen demand, which is an indirect measurement of the organic matter in the wastewater. The RAS is going to meet the influent, and we'll talk about what RAS is in a second. It will either meet it in the same pipe, I've seen it this way, or it'll have its own separate pipe going into typically the front of aeration. Okay, these bugs are hungry, they're ready to eat, and they need dissolved oxygen, and we're going to talk about how this all links up with DO and BOD here in a second. So you're going to have a diffuser down below, supplied by a blower probably, some air delivery system, and it will diffuse the air into the mixed liquor, okay? And what's mixed liquor? That's all the mixed solids in aeration. If you go back to the beginning of the video, you'll see all that churning going on in aeration. That's the mixed liquor, okay? So the air has two purposes. One is to supply dissolved oxygen so that the microorganisms can do their job and consume the organic matter. That is the oxygen demand that they have to do their work, and so we give them that oxygen. The other thing it does is it mixes everything up, keeps everything nice and mixed, okay? And this can be coarse air, fine air, or nano bubbles. Now I'm hearing they're coming out with these small little bubbles. And the smaller the bubble, typically the better the oxygen uptake. And so we use fine air diffusers here. You have a better oxygen uptake, meaning less power bills, okay? And better treatment. So if you were to do a DO profile at the beginning of aeration versus the end of aeration, you will typically see the dissolved oxygen will be lower at the beginning and higher at the end using a plug flow system. Because a lot of the action's happening up here. Hungry bugs, they need air, they need food, and they get to work. And by the time we get to the end of aeration, most of your BOD, if not all of it, should be resolved. Now, when it comes to the different stages, I like to say that in aeration, we resolve the BOD. In the clarifier, we resolve the TSS. So we just talked about how we resolve that BOD. The microorganisms consume the matter, and then they have a dissolved oxygen surplus towards the end. So then it transfers over, and this is a constantly flowing system in a circle, and you always have influent coming in. So there's this aeration basin's constantly flowing, okay? And it's moving to a different process. And so we'll be going over to now the clarifier. In the clarifier, what has occurred in aeration is these microorganisms have created what's called floc, okay? And so there's young sludge, there's old sludge, and typically younger sludge is going to be light and fluffy, hard to settle. The old sludge will be really compact and easy to settle, but so small that it might leave some pin floc behind that floats, and you can't get it to settle without chemical assistance, and they would end up leaving the plant in the effluent. Or if you have it Goldilocks, and this is wastewater treatment just right, Goldilocks. you should have an incredibly clear effluent on the top, or the supernatant actually. It's not the effluent until it leaves in the clarifier, and then you should have a really well compact but not super oxidized sludge at the bottom of the clarifier. Okay, and that's where your TSS is resolved. That flock kind of just grabs all those solids to itself, and I use this, it's adsorption, but for the sake of this conversation, let's say it just kind of creates a net and pulls it all down with it, and it leaves this really clear water on the top. Okay, that is it in a nutshell, but now we have to deal with this sludge, okay? So these microorganisms are reproducing. All right, so we've talked about the return activated sludge briefly, and all that is the RAS is taking that sludge and putting it back into the front of aeration, just like we talked about at the beginning of this process. But if you just RAS all the time and they are reproducing, what will happen is this clarifier will eventually fill up, and it will have nowhere to go, and the sludge will eventually just leave the plant. The sludge, the MLSS, has a BOD load to it. Okay, so you don't want it leaving because you're, in essence, not accomplishing anything at that point. So what you would do is you would, through one way or the other, like I have a separate tap into our area where the RAS is pulled from to run my WAS pump, my waste activated sludge pump. You could come off the same line. I've seen that where you just valve it a certain way and then you can waste off the RAS line. But in essence, you would WAS waste activated sludge to solid handling and get those bugs out of there. This is how you manage your biomass, okay? And I would actually argue the two most important control parameters that I can think of are dissolved oxygen and aeration and your wasting rate, managing your biomass. Of course, pH, temperature, all these things, RAS rate, they're all important, but I would say the operator's two biggest tools are dissolved oxygen and WAS. And so I have seen some operators try to resolve problems with the RAS by turning it up or turning it down. That is not a long-term solution. Sure, in the short term, you took your ibuprofen for your headache, okay, but if you've not, fixed the underlying problem, the headache might return, okay? So anyway, that is how you would handle your biomass, and you would waste out those excess solids. And in those solids are inert as well, and these are things that are going to go to digestion, things that colloidals that might not have been necessarily consumed by the biology, but it certainly was trapped up in the flock by the biology, and you can go take care of that in digestion. So anyway, let's talk about some other vocabulary, and we're going to wrap this thing up. We've already talked about the MLSS, that's the mixed liquor suspended solids. This is an indirect measurement of your biomass, okay? Indirect. You don't hear a lot of people asking when you come to the plant, engineers and operators, what's your MLVSS? You hear them ask about your MLSS. That's really what we use. The MLVSS is the actual measurement of your biomass. That's done, we'll talk about those laboratory procedures at another time, but you need to know this number. It's typically 75% or more. You can have lower, but it's typically 75% or more of your MLSS is MLVSS. The rest of it's going to be inert. That's the stuff that can actually do stuff to the BOD. You need this number for the food to microorganism ratio, or as I like to call it, the BOD to MLVSS ratio. It's the same thing, pounds of BOD versus the pounds of MLVSS in the plant, and that'll tell you something. We can do that math problem at a later time. If you're watching this video and you look in the links and you see the math problem's not there, I just haven't done it yet, but eventually I will, and I'll link to it. Sludge volume index, this is a great tool for using your 30-minute set solids test versus your MLSS concentration, and you can tell kind of how well your sludge is settling. And I'm going to do a video on this. I'm going to do a demonstration with a time lapse, and it's going to be really good. Do a 30-minute set solids, and then we'll do some math. Sludge Age, MCRT, and SRT, these are all used interchangeably, but they shouldn't be. The Sludge Age, I did a video on this. I'll put it in the links below. The Sludge Age is, we're measuring the amount of MLSS in aeration versus the amount of TSS in the influent, like what's coming into aeration. I don't care for this process control because the way, like I've already said, that you manage your biomass is through wasting. Well, MCRT and SRT uses the wasting rate. So I think I prefer to use something that gives me a tool to do something about it. Sludge Age doesn't really. It's still a useful tool, and if you use it, I'm not knocking it. I'm just saying I don't care for it personally. MCRT in California is what we would typically consider the gold standard process control. And so this is using the MLSS in aeration and the secondary clarifier, the entire secondary system, versus the effluent TSS and the WAS wasting, the TSS you're wasting out of the plant in the WAS. And I did that math problem already. It'll be in the links below. And then the SRT is just like the MCRT, except you do not use the secondary clarifier. There's wisdom in this because honestly, you shouldn't have a whole lot of inventory in your secondary clarifier to begin with. People that run high sludge blankets in their secondary clarifiers run into a lot of problems. And so honestly, I see why you would want to use the SRT versus the MCRT. It's a little bit more to the point, but the MCRT actually gives you a better picture, like more of a what is all going on in the secondary system. That about wraps it up. I hope this helped. I hope this gave you a little bit of insight into conventional activated sludge. I know it was a lot, but I want to give you a strong foundation. It's only going to get better from here. It's only going to get more involved. And this process, this is going to take me months to get all this put together because there's just so much we do with activated sludge. It's so involved. But anyway, like I said, if you got anything out of it, like, subscribe, pass to your friends, and we will catch you guys in the next one. Have a great day, everybody.